Hello, my name is Desmond Boykin, and my experiment is the effects of the casein biopolymer in conjunction with activated charcoal to remove contaminants in well water. My question was, can the casein biopolymer remove what activated charcoal fails to remove when filtering well water? I hypothesized that if I use the casein biopolymer in conjunction with activated charcoal, then it will have a synergistic effect in removing the contaminants of well water. The purpose of this experiment is that I am a member of the Nanticoke Indian tribe, and our tribe, along with many other indigenous communities across the continent, face problems with water contamination. So I wanted to find an effective way to remove contaminants in water that activated charcoal fails to remove. You may be wondering, what is casein? Casein is a protein found in milk, and it comprises 82% of all the proteins found in milk. It is commonly formed in the form of micelles, which comprise of alpha S1 casein, alpha S2, beta, and kappa casein. The structure of each of these individual casein biopolymers are still under debate. The composition of casein micelles. Alpha S1 casein contains two polar hydrophobic ends and a hydrophilic center. Alpha S2 casein has two dipolar ends, and beta casein is present as a random coil in an aqueous solution. Kappa casein is predominantly located on the outer surface and comes in hair-like wisps on the outside of the micelle. The purpose of casein is to entrap calcium phosphate inside of milk, so each of these clusters you see are trapped calcium phosphate inside of an entire casein micelle. What is activated charcoal? Activated charcoal is essentially charcoal that has an increased porosity through the use of acids to eat away and increase the surface area. Activated charcoal is commonly used as a well water or a water filter. Activated charcoal removes many heavy metals, but it does not remove any healthy minerals in water, and it also does not remove microbial contaminants such as E. coli. I believe casein will be able to filter out what activated charcoal fails to filter out because through the coils of the beta casein polymer, it may be able to physically entrap any contaminants. Kappa casein will entrap any calcium phosphate in the water. And there is an amino acid in alpha S2 casein that inhibits the growth of E. coli. So casein has antimicrobial properties, which may be able to stop E. coli from being filtered through the well water. The materials for this experiment mainly include casein, activated charcoal, a vacuum filtration system, and a well water testing kit, along with E. coli. The vacuum filtration system, I connected the base to the top by screwing it on and attached a vacuum tube to the filtration system. I then poured 50 milliliters of casein polymer into the filtration system, separated it with a filter paper, then 50 more milliliters of activated charcoal, then I placed a filter paper on top. To test each of the filtered samples, I used a well water testing kit, and the procedures for testing each of the different contaminants varies depending on the contaminant. So I just followed the instructions, which mainly consist of dipping a strip into a sample for a certain amount of time, and then assigning the color the strip turns to a color chart. Here are the results of my experiment. So the pH and the nitrite content. For the nitrite, it started at 15 parts per million for the water control group. And when the water was filtered through just charcoal, it dropped to 4.5 parts per million, while when the water was filtered through casein, it dropped to 3 parts per million. When both charcoal and casein were used, it dropped from 15 to 2 parts per million. The pH did an interesting thing. It started at a pH of 5. When filtered through charcoal, it went to 5.75, and when filtered through casein, it went to 4.25. For the hardness, the hydrogen sulfide, the water control started at 0.2 parts per million. When filtered through charcoal, it dropped to 0.1 parts per million, and through casein, it dropped to 0 parts per million. For the nitrite content, the water control group started at 0.2 parts per million. Through charcoal, it dropped to 0.1 parts per million, and through casein, it dropped to 0 parts per million. For the manganese content, the water control group started at 0.4 parts per million, and the charcoal filtered it to 0.2 parts per million, while the casein filtered it to 0.0875 parts per million. For the alkalinity, the water control group started at 270 parts per million, the charcoal decreased it to 180, and the casein to 80 parts per million. For the cell count, it started at a non-filtered 2,296 E. coli cells, which is a diluted solution of 6 microliters per 100 milliliters. And when it was filtered through charcoal and casein, it dropped to 1,277 cells. This is a 44% decrease in the amount of cells. So in conclusion, it was hypothesized that if powdered casein was used to filter out contaminants in well water, then it would successfully remove the contaminants that activated charcoal failed to remove. This hypothesis is supported through my results, mainly in the nitrite and the E. coli content, as well as the alkalinity. And this experiment could be improved by building a physical body to my filter so it could be attached to faucets, as well as exploring the specific filtration properties of each of the individual casein polymers, alpha S1, S2, beta, and kappa casein. Here is my bibliography, and thank you for watching.